Honors Biology, second quarter review, part 2. Remember, the whole point of making those sugars, for the most part in plants, in addition to making things like cell walls and other structures, is to make ATP by cell respiration. And this is the reason why we talk about things like high blood sugar, low blood sugar. When you have low blood sugar, that means you have less sugar to break down in the mitochondria by cell respiration, which means that you're going to be making less ATP, less energy, and that's why you feel tired. So let's talk about that really quick. We have ADP, or adenosine diphosphate, and basically we have two phosphates attached to this adenosine part. And don't worry about this adenosine, but and even these first two phosphates aren't really that important for what you need to know. But without that third phosphate, which is going to be held on by a very unstable bond, which is good because you're going to be able to transfer it to another molecule very easily, this ADP doesn't have any energy that it can get. It's too stable. So I want you to think of ADP as like an uncharged battery or a partially charged battery. It doesn't have energy to provide to other chemical processes. Now, the whole point of that cell respiration process in the mitochondria is to add the phosphate back on the ADP to make ATP. Now, this third phosphate is not held on very tightly. It's very reactive, and that's one of the reasons why you can't store it. It's too reactive to store. You store sugars, you don't store ATP. This ATP it has energy. It can provide that energy to other chemical reactions, and the way it provides energy is the loss of the third phosphate group, and it's going to be leaving that ATP attached to another molecule, destabilize it, change its shape, and make it do something. So ATP works by losing a phosphate group, the third phosphate group. And then what does the mitochondria do? Add another phosphate to it, using the energy from sugar. Here's an energy cycle. We have the twin processes of photosynthesis and cell respiration kind of cycle together here. Photosynthesis uses CO2 and water, using light energy, to make glucose and oxygen, oxygen being a waste product. However, oxygen is necessary for cell respiration. That's why you're breathing in oxygen now. And then we also have glucose being broken down, glucose and oxygen in cell respiration, the mitochondria, broken down to make ATP energy, and producing CO2 and water as waste products, which again could be used for photosynthesis. So where does this stuff happen? Well, in the plant, for photosynthesis, it happens in the leaf. Basically, that's the food-making factory. Now, for things like cacti, they do their photosynthesis in the stem, but basically the green parts of a plant. So we have the very top layer, it's waxy covering, called the cuticle. That just prevents water loss. It's kind of like a protective layer, like wax paper. Then we have a layer of cells called the epidermis, basically the skin of the leaf. This is a cross-section of the leaf here. Again, that doesn't really involve anything with photosynthesis, but it does involve protecting the inside of the leaf from infections, fungi, things like that. The part that's going to do most of the photosynthesis in the leaf is the middle part. This middle part is called the mesophyll, and meso means middle. This part is going to have a lot of chloroplasts, a lot of places to do photosynthesis. We have the palisade mesophyll that you don't have to know for honors biology, and we have the spongy mesophyll as well, and they're just places we're going to do a lot of photosynthesis. Here we have a vein in the leaf. We bring up water from the roots through the veins, and you can see the veins there, just kind of like our veins, you transport things around. And then sugars made by photosynthesis are transported down through the veins to the rest of the plant, and extra sugars are stored in the roots. At the bottom, we also have a epidermis as well as a cuticle, and also in the bottom are these little holes that lets gases in and out. These holes are called stomates. Stomates are the holes, and there's two little guard cells guarding it, kind of opening and closing depending on the conditions outside. If it's dry outside, they close. If it's nice outside, they open. Here we have the water vapor that was brought up from the roots through the veins, called xylem, and eventually it's going to evaporate out. However, about 1% is used in the process of photosynthesis. Remember, you need water, sunlight, and carbon dioxide to do photosynthesis. We also have oxygen being made in photosynthesis. That's how the oxygen is going to get out as well, through these little holes called stomates. And this is how we get carbon dioxide into the plant. Now, keep in mind, if it's dry out, then these stomates close. You can't get carbon dioxide into the leaf. Plant can't do photosynthesis. And that's going to be a bad day for the plant. There are some plants that have some tricks, like CAM and C4 plants, that can do their photosynthesis parts in different times of the day or different structures, but that's not the focus of this class. You should know, however, that photosynthesis happens in the chloroplast within the cells of the middle layer of the leaf called the mesophyll. Here we have the pigments of photosynthesis. The chloroplast is the part of the cell, the organelle inside the cell, that does photosynthesis. Inside the chloroplast are these stacks of little pancakes called thylakoids, and the thylakoids are going to have embedded a very tiny molecule called chlorophyll. Chlorophyll is the light-capturing pigment inside the chloroplast. Don't get that confused. Chloroplast, much bigger than a chlorophyll molecule. Basically what's going to happen here is the chlorophyll is going to capture light energy and transfer that energy to electrons that are going to be used to make some high-energy molecules in light reactions. Remember, chlorophyll is a light-capturing pigment, and if you notice it's green. This is the reason why plants are green, because of the chlorophyll. The light hits the chlorophyll, and then all the colors are absorbed except for green light, which bounces off, and that's why plants appear green. Here we have a photosynthesis overview. You should know that photosynthesis is broken down into two parts: the light reactions, that, as the name implies, only happens in light, and the Calvin cycle, which doesn't require light. So we have the light reactions here happening in the thylakoids of the chloroplast within the plant cell. Light is captured by chlorophyll. We have water coming in that's going to be broken apart in the light reactions. And then as a result of breaking apart the water, we're going to release oxygen. Remember, that's one of the waste products of the process of photosynthesis. Now, the light reactions are also going to produce two more things, two high-energy molecules called ATP and NADPH. These just have energy, and they're going to provide energy to the Calvin cycle, which is the second part of this process of photosynthesis. So part one, light reactions, thylakoids, light coming in, water coming in, oxygen leaving as waste product, ATP and NADPH being made. Calvin cycle, second part. Calvin cycle happens in the liquidy interior of the chloroplast called the stroma. And inside the stroma, we have carbon dioxide coming in from those stomates. Remember, those were the holes in the bottom of the leaf. And it's going to diffuse into the stroma. Using the energy of ATP and NADPH, we're going to attach some hydrogens to that carbon dioxide and rearrange it a little bit, and eventually turn it into sugar. In fact, if you remember, the formula for sugar was C6H12O6. The C's and the O's came from carbon dioxide. So the Calvin cycle is taking gas, carbon dioxide gas, using energy that ultimately came from the sun, and the plant's going to put together its sugars. Now, these sugars they have stored energy for cell respiration in the mitochondria, as well as these sugars can be used for building up things like cell walls, which are just basically chains of sugar called cellulose. Review what goes in and what goes out of the light reactions and the Calvin cycle and where it happens. Here we have cell respiration. This gets a bit more complicated. Remember, that happens in the mitochondria. The first part of cell respiration is called glycolysis. Glyco means sugar, lysis means break apart, and that's what we're doing. We're breaking apart the sugar. Glucose comes in, ultimately came from the foods we ate, and we're going to bust it apart for some energy, making something called NADH, which is going to transfer some high-energy electrons from the sugar to the last step called the electron transport chain. After we bust our sugar in two, we now have two half sugars with some of its electrons missing called pyruvate. And in this process, we're going to make a couple of ATP, only two, which is not a lot. Keep in mind, things like bacteria don't have a mitochondria, so all they do is make two ATP in their cytoplasm. The two pyruvate enter the next step, which is in the matrix, or the center part of the mitochondria, and enter something called the Krebs cycle, which is just a cycle of chemical reactions catalyzed by enzymes to further bust apart that sugar for energy. In the Krebs cycle, we're going to make two high-energy molecules, NADH and FADH2. There's a total of 6 of those NADHs being made, 2 FADH2s being made. And again, we're just ripping apart that sugar for energy and transferring the energy using these transporter molecules. And they're going to drop off that energy at the electron transport chain in the form of high-energy electrons. In this process, we're going to make 2 ATP, not very much, and by the time we're done with the Krebs cycle, there's no more sugar left. It's been completely busted apart into carbon dioxide gas. The carbon dioxide diffuses from high to low concentration into your bloodstream, and then eventually diffuses into your lungs, and that's what you breathe out. The last step of cell respiration is electron transport. And what we're going to do is we're going to take the energy from NADH and FADH2, which ultimately got their energy from sugar, And using that energy, we're going to generate a whole bunch of ATP, 34 ATP total. So without a doubt, electron transport produces more energy than either of these two other processes. Also, the electron transport chain is going to use oxygen. This is why you're breathing oxygen right now. The oxygen is going to accept these electrons from these two molecules, and I'm not going to test you on that. But basically the oxygen is going to accept 2 electrons, 2 protons, which is 2 hydrogen, and turn that oxygen into water, H2O. And this is where the water is produced in the cell respiration formula. If you remember, we have glucose plus oxygen yielding ATP plus water, which is coming from electron transport, plus carbon dioxide, which comes from the Krebs cycle. If you're looking for an advance on the exam, then you do need to know this. Alright, cell cycle, going from 1 cell to 2 cells. Not birth to death of a cell, but 1 cell to 2 cells. The interphase part of the cell cycle is the cell doing its everyday job. And then we have a mitotic phase where the cell is actively dividing. Interphase, inter means between, so this is like between cell divisions, is divided into 3 parts. We have G1, stands for gap, but you can kind of think of it as growing and doing its everyday job. If you're a liver cell, you're detoxifying. If you're a muscle cell, you're contracting, all that good stuff. During the S phase of interphase, the chromosomes are being copied. Now every chromosome is going to consist of 2 sister chromatids attached at the centromere. And of course we're going to need 2 copies because during the mitotic phase we're going to separate them out during anaphase. The last part of interphase is called G2. And again, the cell's growing and making new organelles, preparing for cell division. So that's interphase. And then after interphase, G1, S, and G2, we enter the mitotic phase, which consists of mitosis as well as cytokinesis. So we are going to do the 4 phases of mitosis, which is prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes become visible and the nuclear membrane breaks down. During metaphase, that's an easy one to remember, think middle phase, the cell's chromosomes line up in the middle. Then during anaphase, the sister chromatids separate and they move to opposite sides. And then during telophase, we have 2 nuclei forming, and after that, we have our last step after mitosis, which is cytokinesis, and then the rest of the cell divides in 2. That's how we go from 1 cell to 2 cells. And now the cell enters into G1 of interphase again. Remember, this happens for all body cells. Cancer and cell growth. Remember that cancer is essentially a failure of cell division control. And we're just talking about uncontrolled mitosis. Normally your cells aren't just dividing over and over and over again uncontrollably. In cancer they do, and that causes like a tumor. Now, as far as details, you don't have to know this for this class, but there's a gene that regulates cell division, and when that gene is broken and some other things happen, then the cell will basically just keep on dividing, and that's what cancer is all about. If the cancer doesn't spread, then it's very treatable. You just cut it out and life goes on. However, if the cancer spreads or metastasizes, then the cancerous cells will make exact copies of the broken genes for cell division control and start cancer in new areas of the body. How do we make sperm and egg? Well, we're going from 46 chromosomes to 23 chromosomes. Basically, we're doing a different type of cell division called meiosis. Remember, during mitosis, we go from 46 to 46. Meiosis, making sperm and egg, gametes, goes from 46 to 23 chromosomes. When the sperm and egg fuse together, now we're back at 46 chromosomes in the zygote, and now this cell will undergo mitosis. So meiosis, just making sperm and egg, going from 46 to 23. Now keep in mind that's for us humans. If we're dealing with a different organism, saying an animal that has 41 or 40 chromosomes, then by meiosis it would make sex cells with only 20 chromosomes. Here we have a review of mitosis versus meiosis. In mitosis, body cells, somatic cells being made, we have 1, 2, 3, 4, and at the end we end with 1, 2, 3, 4. So we have identical copies of the DNA being made in the process of mitosis. During meiosis, we have 2 rounds of cell division, and we're going to make a total of 4 cells. In mitosis, it was just 1 cell division and 2 cells being made. During meiosis 1, we're going to separate out the homologous chromosomes, and they're going to be packaged into different cells. And then during meiosis 2, the sister chromatids get separated, and now every cell, every sex cell, every gamete will have 1 copy of every chromosome. Here we have 2 copies of every chromosome in mitosis, 1 copy in meiosis. Diploid, 2 sets, ending with diploid in mitosis, starting with diploid in meiosis, ending with haploid cells in meiosis. At this time, we will stop our video, and this ends part 2 of your review.